Hello and welcome back to the AI Research Roundup. I'm your host, Alex, and today is May 6th, 2025. Alright, we're looking at this ICLR conference paper published just over 3 months ago, titled Steering Protein Family Design Through ProfileBFN Flow. This paper introduces a method called ProfileBFN. It's designed to generate diverse, functional protein sequences within a specific family, but cleverly avoids the heavy computation usually needed for large multiple sequence alignments, or MSAs. Okay, so building on that introduction, this table compares ProfileBFN against several other models using data from Cameo. It assesses performance using sequence metrics, like diversity and novelty, and also structural metrics, things like long-range contact prediction accuracy. Well, the key takeaway seems to be that ProfileBFN, particularly the profile-guided version, really excels, often outperforming competitors like DPLM and even the baseline searched MSA on those structural measures, showing it captures family characteristics well. Okay, moving on. This figure really visualizes that point about capturing structural characteristics. We see 3 examples of lysozymes generated by ProfileBFN, shown in cyan, overlaid with reference structures in pink. Look how closely they align structurally. The high PLDDT and TM scores confirm this conservation. But importantly, the sequence identity remains quite low, showing these are indeed novel sequences. It really drives home the model's capability to generate diverse yet structurally sound proteins. Alright, and this figure provides another angle on that structural accuracy point we just discussed. So here we're comparing protein contact maps. On the left, you see the map generated using ProfileBFN, and on the right, one from a standard searched MSA. Well, look at the long-range P at L scores. ProfileBFN actually achieves a higher accuracy, 51.5 compared to 39.7. It suggests ProfileBFN captures these crucial long-range interactions really effectively. So beyond just structural accuracy, this next table dives into functional performance, specifically on enzyme tasks. It compares ProfileBFN against POET MSA and EvoDiff MSA using an accuracy times uniquenesses metric across 3 different enzyme families. Well, the results are pretty stark. ProfileBFN achieves much higher scores, suggesting it's far more likely to generate enzymes that are both unique and actually perform the intended function compared to the others. Okay, so besides that strong functional performance we just saw, how fast is ProfileBFN? Well, this plot compares sampling time against sequence length for the different models. You can see ProfileBFN, those orange and red lines, stays pretty low. This means it generates sequences much faster than competitors like DPLM or EvoDiff, particularly as proteins get longer. So it's not just effective, it's also quite efficient computationally. Alright, so we've seen it's fast, but how well does ProfileBFN actually understand proteins beyond just generation? Well, this table, Table 3, looks at performance on various prediction tasks, things like thermostability, metal ion binding, and localization. Across the board, ProfileBFN consistently performs really well, often beating other protein language models and its diffusion competitor, DPLM. This suggests it learns a really deep, biologically relevant representation of proteins. Okay, so that representation quality we just saw in Table 3 really sets the stage for this figure, Figure 5. Here, we're comparing ProfileBFN, shown in blue, against POET and EvoDiff for generating phage lysozymes. Well, the plots show sequence novelty versus predicted structure quality, measured by PLDDT and TM score. You can see ProfileBFN consistently generates structures with high confidence and similarity to natural ones, while still maintaining good sequence diversity. So, wrapping things up, ProfileBFN looks like a really strong method. It effectively generates diverse, functional proteins that maintain family structure, and crucially, it does this efficiently, even training on single sequences. The strong performance across generation, prediction tasks, and speed really makes it stand out. Well, that's all for this time on the AI Research Roundup. Thanks for listening. I'm Alex.